Okay. Do you guys, I'm curious, do y'all get a notification when I start recording?
Yeah, and then it asks us like to continue.
Oh, okay. I was wondering if it let you know when I'm recording. Okay. So today we are in chapter 12. Check Blackboard for Codon Dictionary. I sent it to the chat window. If you didn't get the Codon Dictionary, let me know and I can send it to you in Blackboard. Let me just send it to everybody right now. That way I know you have it in Blackboard. I'm going to send it as a PDF. Okay. I'll send it real quick. And so if for some reason you didn't get the Codon Dictionary in the chat window, check Blackboard, your course messages. I just sent it right now, so it should be there for you. Okay, so chapter 12, we're going to talk about what's called the central dogma of genetics. The central dogma of genetics. And this word dogma right here, okay, this is like core beliefs. Okay, it's like the central dogma is like the core, the most shared consensus among all scientists as to what is it that DNA does for us. And I'll write it out in words and then I'll write it out in a diagram because it's depicted both ways. But the central dogma of genetics says this. First of all, DNA is the molecule of heredity. Okay, that's something that you already know. Okay, this is the genetic information of all living things. DNA copies itself through a process called DNA replication, which is what we talked about last time. And the enzyme responsible for DNA replication is DNA polymerase. So DNA copies itself through a process called DNA replication, and the enzyme responsible is called DNA replication. Then here's kind of the new parts. DNA is used to make a coded copy of a gene called messenger RNA. So DNA is used to make a coded copy of a gene called messenger RNA. The process is called transcription. And the enzyme responsible is called RNA polymerase. So DNA is used to make a coded copy of a gene. The enzyme responsible is called RNA polymerase, and the process is called translation. The last part of the statement is that messenger RNA is a coded copy of a gene. Okay, messenger RNA is a coded copy of a gene. It's decoded by a process called translation. During the process of translation, transfer RNA and ribosomal assemble amino acids into a protein. And so this one, basically these guys right here, they are the enzymes in a sense that do this. They're the enzymes, transfer RNA and ribosomal RNA. In a sense, they're the enzymes. So this is a lot of detail. Type a 1 in the chat window when I can draw my diagram, because I want you to watch me to draw the diagram so that you can kind of make sense of what we just wrote. So let me know when you're ready for me to draw the diagram. Type a 1 into that chat window. Good, good. Y'all are keeping up. Good. So here's the diagram, and I'm going to use 2 different colors to draw out my diagram. But here's what the diagram looks like. We start out with DNA, RNA, and then a protein. So in the second statement, we said that DNA copies itself. So we draw a circle that kind of points to DNA because it copies itself. And we say that process is called replication. And the name of the enzyme responsible is DNA polymerase. So DNA makes more DNA through the process of DNA replication, and the enzyme that does that is called DNA polymerase. Then 3 and 4, this is what we're going to spend the rest of our lecture time on. DNA is then used to make something called messenger RNA, or sometimes we just abbreviate it mRNA. The process of using DNA to make mRNA is called transcription. The enzyme responsible for transcription is called DNA polymerase. mRNA is used to make a protein. The process, and I was going to underline these, the process of going from mRNA to a protein is called translation. The enzymes responsible are tRNA, which is the transfer RNA, and rRNA, or the ribosomal RNA. I guess I could have underlined these too, because you're going to see their names in there. And the end result is a protein. So this diagram basically states these four statements. On page 208 of your textbook, and I'm going to share this with you, but go ahead and copy my diagram first, because this diagram's not in your book, but I'm going to show you which diagram is in your book. Let me know when you've copied this diagram, and then I'll switch to your book real quick. So type in number 1 in the window when you've copied this diagram. Good, good. Okay, so on page 208 of your textbook, they've greatly simplified this diagram, and they simply show it like this: DNA to mRNA to a protein. And then, of course, they have transcription and translation. Okay. And so let me show you how they represent this in your textbook. But they've simplified this. And right here in the middle of the page, they have it nicely stated, said briefly. Oh, I forgot I'm using your book. Said briefly, DNA is used to make RNA, is used to make a protein. And so here's the representation of DNA. Here's transcription is used to make mRNA. Here's translation is used to make a protein. So that's going to be the topic of the rest of our lecture today, is what is transcription and what is translation. So if you're following along in your book, we're starting here about page 208 to page 209. Okay. So before we jump right into transcription, let me give you a little bit of a reminder about DNA. Okay, DNA a little bit. Remember that we said there are two sides to the ladder. Okay, there are two sides. One side is referred to as the coding strand. The other side is the non-coding strand. The coding strand has other names. Okay. The coding strand has other names. Sometimes it's also called the sense strand. Um, or sometimes it's referred to as the gene strand. But all of these terms, coding, sense, and gene, they all refer to the same side of the DNA strand. The coding strand, or the sense or the gene, this is the side that actually contains genetic information. This is the side that has your actual genetic information. And what is your genetic information? That is the sequence of bases. You know, we hear all the time, that's your genetic information. Well, what is it exactly? Well, the genetic information is literally the sequence of bases. So if this is an A, a T, a C, a G, okay, it's the order of the letters. That is your genetic information, the order that they go in. The other side, there's bases over there, but those bases don't make any sense. In other words, their order is not what determines anything about us. The only purpose of the letters on this side of the strand is to cover and protect the other strand. So the non-coding, it has bases, and the bases are complementary, but this is not the side of the strand that we pay attention to when we're looking for your actual genetic information. We only pay attention to one side of the strand. So the non-coding side is sometimes called, and I promise I'm not making this up, the nonsense strand. It's actually called nonsense. Or sometimes, instead of being called the gene, it's called the complement strand. And I was looking in your book real quick. I can't remember what they call it. I think they call it the complement strand. But if you go to other videos online and you hear them using the words nonsense and complement instead of sense and gene strand, I want you to know all the possible names. Yeah, all the possible names. But the function of this side, it covers and protects the bases. So yeah, there's still, like I said, there's still, oh, I have to fill in the complementary. T-A-G-C, T-A. Oh, I can't tell. I think this side is shorter. C-G, there we go. A. So they're still complementary, but when we are looking for a sequence, we're looking at this strand only. So now I'm going to draw another DNA ladder, this time laying on its side. Okay, so here is the gene strand. This is the complement. Remember that the gene strand is the same thing as the coding side. When we talk about genes, your genes are a certain area on the gene strand. So it might be literally like 100,000 bases, or it might be only like 50,000 bases. Some genes are very long and take up a long space, and some genes are relatively small and don't take up a lot of space. But when we use the word gene, we're talking about some bases, the order of the bases that are on one side of your DNA only. So this is a section of the gene strand. That's what a gene is. It's one small section of the gene strand. So you don't find genes on both sides of your DNA. You only find it on one side of your DNA. So this idea that we're only going to be using one side of the DNA and that the genes are only one side, that's an important concept as we start talking about transcription and translation. I'm going to show you a short video that's actually from your textbook. Before I show you the video, type a number 1 so that I know that you have this diagram because it's not in your book. So show me a number 1 in the chat window so that I know that you have this diagram copied. Oh, sorry. Okay.
Oh, Miss Weir, I can't hear you.
Yeah, me neither.
Okay, can you hear me now?
Yes, now I can.
Can you guys hear me talking now?
Yes, we can, or I can.
Yes? No? Can you hear me talking now? I'm guessing you can't hear me talking now. Yeah. I think this is a problem with this switching from my headset back to okay, so you can hear me talking. Okay, so you can hear me talking. Okay. All right. Well, I'm sorry about that. I thought I had fixed the problem with the video sound. I think the issue is when I use my headphones and I switch to video, there's something about my computer settings that it doesn't switch the sound back to my computer. So, okay. Well, then good thing the video was only 2 minutes long and it wasn't an hour long. That would have been terrible. All right, that's okay. We can go on without the video. But y'all can hear me now, correct? Type a 1 if you can hear me speaking now. All right, good. Thank you, Nancy. Okay. All right, then. Well, then we won't worry about the video. I'll post a video link in Blackboard that will go with today's topic. And we'll go ahead and we'll finish this without the video. All right. So here we go. Transcription. All right, so the process of transcription. We're going to draw a diagram and then also write out the steps. So step 1 is that the DNA has to unwind and lay flat and then begins to unzip in the area, and this is, I'm going to underline this, in the area of the gene. Okay. I'm going to underline this because this is a little bit different from DNA replication. It's going to unwind and unzip in the area of the gene to expose the bases. And in the second step, the enzyme RNA polymerase binds to the gene strand and adds complementary RNA nucleotides until entire gene is copied. And I'm going to underline the word RNA polymerase because that's important. Gene strand, RNA nucleotides. And then we'll draw a diagram to go with this. So in the first step, our DNA ladder has to unwind and lay flat, and then we're going to add RNA nucleotides until the entire gene strand is copied. This is going to be the complement strand. This is going to be the gene strand. And I'm going to give myself a sequence. T, A, C, G, 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 C, T, G, A, G, A. Okay, give myself a little sequence on that side. So here comes the large enzyme, which I'm just going to represent by this big pink fluffy cloud. Okay, this is the enzyme. This is RNA polymerase. And it's going to use this side as a pattern to build a strand, but not of DNA, a strand of RNA. So half of our picture is going to be DNA and the other half of our picture is going to be RNA. So across from here, we're going to have not DNA, but RNA nucleotides. The pairing rules from DNA to RNA is when DNA has adenine, across from it, it's going to contain what's called uracil. When DNA has thymine, it's still going to take A, and the C and the G rule stays the same. So one difference between DNA and RNA is that DNA has no thymine in it. Instead of thymine, we substitute a base called uracil. So normally across A, we would have a T. But since we're building RNA, instead we're going to have a U across from it. So here's my mRNA strand. My other pairing rules are still the same. So G's and C's pair. My other pairing rules are still the same. But wherever I would normally have a T, wherever I would normally have a T, instead, I'm going to have a U. So one difference between DNA and RNA is that RNA doesn't have a U in it. Or RNA doesn't have a T in it, sorry. RNA doesn't have a T in it. So if you look at my diagram, I do have a triple strand of DNA. There's the complement, then there's the mRNA, and then there's the gene strand kind of sandwiched in between. The enzyme, notice, is not paying attention to the complement strand. It's only paying attention to the gene strand. It's only paying attention to the gene strand. So some differences between our messenger RNA and our DNA. The nucleotides that we have available are adenine, uracil, cytosine, and guanine. Remember that uracil replaces thymine. DNA has thymine. But RNA doesn't have thymine. The way these nucleotides are built is that you still have a sugar, but the sugar is ribose. In DNA, the sugar is deoxyribose. You still also have a phosphate, and you still have a base. And remember, your bases are adenine, uracil, cytosine, and guanine. So an RNA nucleotide is still a sugar, a phosphate, and then one of the 4 bases. So it has the same general shape. It's just that in this case, the sugar is going to be ribose instead of deoxyribose. And the bases are going to be A, U, C, and G. So there's a little bit of difference between an RNA nucleotide and a DNA nucleotide. The difference is the sugar, and the difference is one of the bases. So when we're building this new strand, there is ribose on this side as part of the backbone, but there's deoxyribose on the other side as part of its backbone. So as we're trying to show in our drawing, and as we stated, the enzyme, it's going to bind, pay attention to only one side of the strand, and it's going to add along the strand RNA nucleotides until it completely copies the gene. So until it completely copies whatever small section of DNA that it's paying attention to. Step 3 is when the gene is copied, then that RNA polymerase enzyme detaches from the DNA. And the messenger RNA also detaches from the DNA gene strand. Oh, sorry, you can't see what I'm writing. Okay. So when the gene is copied, the DNA polymerase enzyme detaches from the DNA, and the mRNA also detaches from the gene strand. The DNA molecule, I guess we could say it rezips and rewinds into helix. So here's your DNA helix rewinding. Here's your enzyme that you have left over. And then you have a small, tiny little messenger RNA. So at the end of this process, here's what you have left over. The result is a small single-stranded RNA molecule that is a coded copy of a gene. So you have a small single-stranded RNA molecule that is a coded copy of a gene. So this little piece is not a copy of the whole chromosome. It's just a copy of a small little piece. Okay. Hey, I think I figured my setting. One of y'all say something out loud so I can see if my headphones are working again. Brandon, do you still remember the Pledge of Allegiance?
Barely.
Barely. Hey, I can remember. Okay, I can hear you now. Okay. I found the setting. I can hear y'all now. Okay, good. What do you mean barely remember the Pledge of Allegiance? You said it like every day for 12 years.
I was just kidding. I was just kidding.
Okay, so I'm going to practice something with y'all that you're going to see on a quiz. So let me practice something that you're going to see on a quiz. So here's is an example problem. Let's see if you can determine the correct, determine the mRNA sequence from a given gene sequence. So see if you can determine the mRNA sequence if I give you a gene sequence. And let's say the gene sequence looks like this. Okay. So here's the gene sequence. Could you tell me what the correct mRNA sequence would be? So we'll take turns. I think I have one sequence for everybody. We'll take turns. All right, Brandon, you're first. I'm going to pick on you first. If the DNA says TAC, what 3 letters would be on the messenger RNA side?
For TAC,
Mm-hmm.
A, U, and G.
Yeah, very good. A, U, and G. And just as a reminder, if in the DNA you have this, the pairing rules. Okay. Here's our pairing rules. All right, Erica, you're next. If in the DNA we have GAC, what are going to be the 3 letters across from it in the mRNA?
Okay, sorry, I just ate a granola bar. Okay, so
You stayed a granola bar.
yeah, I'm snacking.
I'm dying of slow death over here.
So it'd be C T G.
Not T, C.
Oh, U.
There you go. CU what?
G.
Thank you very much.
Right?
Very good.
Yeah.
Okay, Nancy, the next one is yours. And Frankie, I'm going to let you know, Frankie, yours is the TAA one. Go ahead, Nancy.
It's UAU.
Very good. Frankie, you get the last one. What would be across from TAA? And you can type in your answers if you don't want to say it out loud. What would be yours, Frankie?
AUU.
Perfect. And I'll do the last one. GCA. Perfect. So if you are given a question like this on the quiz, make sure that when you're doing mRNA, you remember that there is U in the sequence. All right, good. So now we'll talk about the last part, and this is where we're going to need your codon dictionary, and that is translation. Now, I will let you know that your textbook gives you way more information than I would expect you to know about translation. So I'm going to give you a simple description of translation. So the first step in translation is that the newly made mRNA leaves the nucleus. This whole time we've been in the nucleus because that's where the DNA lives. That's where the DNA lives. We're going to have to leave the nucleus and enter the cytoplasm because that's where the tRNA and the rRNA live. So the tRNA and the rRNA, they live in the cytoplasm. We need them for the next step. So the newly made mRNA leaves the nucleus and it enters the cytoplasm. There it's going to locate the next two enzymes that we need, the ribosome, which is also made of ribosomal RNA, and tRNA, which is the transfer RNA. The next step is that the ribosome binds and attaches to the mRNA. It binds and attaches to the mRNA. It's going to read the mRNA one codon at a time. And a codon, right here, a codon is 3 sequential bases. So 3 bases in a row is a codon, 3 sequential bases. So the ribosome is going to read the mRNA 3 bases or 1 codon at a time. While that's happening, the transfer RNA brings amino acids to that ribosome. And then the last step is that the ribosome links amino acids together. I guess this is really the last step, and we'll draw a picture. The process continues until you read the entire mRNA. So the process continues until we've read the entire mRNA strand. Okay. So we're going to draw a picture and we're going to use our codon dictionary. Okay, here's our diagram. So we're going to give ourselves a relatively long mRNA strand here. We'll use our same sequence that we used earlier. Okay. So here we'll say we have AUG, CUG, UAU, AUU, GCA, and we'll give ourselves one more, UAA. So this is our mRNA. I'm drawing the ribosome here. This is the ribosome, also made out of, rRNA. It's reading one codon at a time. And then there's a really strange looking, I promise I'm not making that shape up, but there's a really strange looking molecule. That's the transfer RNA. And it's bringing with it an amino acid. It's bringing with it an amino acid. So here's how we know which amino acid transfer RNA is bringing. Let me get my codon dictionary here. Ah, here we go. Okay, my printer's acting crazy, so I'm not going to print it. Let me share my screen instead. Oh, you know what? I'm out of time. I'm out of time as I look at the clock. Okay, we'll finish this after spring break. That means that I'm going to have to adjust the deadline on the chapter 12 transcription translation quiz. Will be due probably the Monday after spring break, April 5th. Okay, so the only thing you have to worry about to be due is the DNA structure and replication quiz. Let me just make them both due April 5th because I hate having to remember multiple deadlines. What do y'all think? Let me just push that quiz back, and then the chapter 11 and chapter 12 quiz due April 5th. Okay. So I'll push everything back I'll push everything back 1 week after spring break so that I can finish this piece of information before you come back because I am out of time. How does that sound, you guys? Does that sound okay with you?
Yes.
Okay. You're going to need, I'll tell you, you're going to need your code-on dictionary to do this quiz. So when we come back from spring break, I'll remind you on how to use it, or I'll show you how to use your code-on dictionary. The other thing is today, I cannot meet with you for lab. I'm so sorry, but I have a huge, huge project that I am working on for my boss, and it is due tomorrow. It is due tomorrow. I have 8 forms I've got to fill out. So I cannot meet with you in lab. I've been using my lab time to work on my project, and I'm not quite halfway done. So I'm hoping to finish it tonight before I go to bed. But I cannot meet with you today so that I can work on that project. But the lab that's posted there this week is not a difficult lab. It's a very simple lab. It's pretty easy to do. But in case you also didn't know, next week is spring break. So no Zoom meetings for next week. The next time I will see you, our next Zoom meeting will be March the 29th, or yeah, our next Zoom meeting is March the 29th. Oh, no, no. Our next meeting is March the 30th. Yeah. Our next Zoom meeting is March the 30th.
And Miss Ware, is there any way I can get the deadlines for the lab class?
Yeah, I'm going to make the deadline for the lab class to also be due April 5th.
Okay, you're the best. Thank you.
Yeah. I like everything to be due on the same day. So whatever was normally due in lab on March the 29th, and I'll post this to Blackboard. I'll post an announcement. I'll move it to April 5th since I'm moving the quizzes to the same date. Okay? You guys have been a good audience. Let me stop recording here. You guys have been a good audience. I hope